Hey y'all, my name is Caroline Lowry, and this is my PCR presentation. So I'm going to tell you a little bit about the PCR process. PCR is also known as the polymerase chain reaction, and you'll hear some people refer to it as this, and you'll hear some people refer to it as the thermal cycler. So basically, you can think of this as a genetic copy machine, which is super cool to me. And in this process, you have to supply the components in a certain order in order for millions of copies of DNA to be produced within hours. When all the components are functioning correctly, the PCR is able to double the amount of target DNA in each cycle that happens. So this is why repetitions take place, because the more repetitions that take place means there's more copies, which is the purpose of this. So now I'm going to talk to you about the components of PCR. There are several components that have to be brought together in a specific order in order for this process to work. The first one being nucleotides, and this provides a starting point for the replication DNA. The next one is an enzyme known as Taq. This amplifies or synthesizes the DNA. It must be added to the DNA template, which is what you want to make copies of. Magnesium chloride and primers are also used. Taq is a DNA polymerase. You don't have to use Taq, but many people, inventors, whatever, that are doing the PCR process prefer this because it is considered thermophilic, so it can work at very high temperatures. It has a half-life of more than 2.5 hours, and it has a high amplification capacity as well, and it can add approximately 150 nucleotides per second, which is a lot. So it's often used and much preferred by everyone because of those reasons. Magnesium chloride, or MgCl2, is essential because it enhances the activity of the DNA polymerase Taq. This results in the amplification rate increasing substantially. However, if you use too much of that, it can result in a decrease in the amplification rate. So you got to watch that because only a certain amount, you're able to use. The primers in the reaction specify the exact DNA product to be amplified. So these are short fragments with a defined sequence complementary to the target DNA that is to be detected and amplified. Primers serve as an extension point for the DNA polymerase to bind on. So I'm going to continue telling you about more components. So the next thing that happens after all these components come together, a thermal cycler, or as some people will refer to it as PCR machine, either one, is much needed to help these components make copies of whatever you're wanting. So this machine raises and lowers the temperature of the thermal block in very precise steps. Then 3 steps take place, the first one being denaturation, which is when the separation of target DNA strands happen. The second one is hybridization, or some people will say annealing, either one. This is the binding of specific primers to target DNA segments. And the third step that will take place is elongation or extension. And this is where the DNA polymerase extends the primers by adding nucleotides to the developing DNA strand. That's so cool to me. So repetition, like I discussed previously at the beginning of the video, repetition of these steps is what takes place multiple times throughout the process, and this exponentially produces the exact copies of target DNA, which just means it produces a ton of copies of whatever DNA strand that you're trying to copy, and this is why these steps are repeated so many times throughout the process. So overall, a basic PCR reaction consists of target DNA, DNA polymerase, which most people will use Taq, or you'll hear about Taq in the PCR process, nucleotides, primers, MgCl2, magnesium chloride, and a reaction buffer. So to detect PCR products, PCR is the first half of the reaction. PCR amplifies the target DNA, which this is what has to be detected. So detection of PCR products is determined by using electrophoresis, which is just the movement of particles in an electric field, if you did not know what that is. Electrophoresis powers a power source in a substrate, such as agarose in this case, for example. Agarose and 2 other substrates can be used, but agarose is the most preferred. It's the easiest, and it's very basic to use, so you'll hear about that being used often. It's a seaweed-based substance, and when it's added to a buffer solution, like a jelly-like matrix is formed, and this is formed whenever it's heated and cooled. The other 2 are capillaries and acrylamide, but you mostly will hear agarose being used. So in real-time PCR, this measures the incorporation of the fluorescent dye into an increasing amount of a double-stranded DNA. It's used to qualify the amount of target DNA in the sample and for the presence of the appropriate DNA sequence. So the overall goal of PCR is it gives you the ability to detect polymorphism by making enough copies of a specific fragment of DNA so that you're able to see it. If you don't know what a polymorphism is, it's a DNA sequence difference between biological samples. So many differences that you can see are called phenotypes, as you may have learned earlier in your school years. An example of a phenotype would be like why you have blonde hair and I have brown hair, or why you have blue eyes and I have brown eyes. It's physical differences that you're able to see and compare from one person to another. Genes that create these polymorphic phenotypes are the genotype of an organism, and every organism has a genotype. So you don't need PCR to determine the phenotypes, but if you see a phenotype, in order for you to determine the phenotype, you might need PCR. So many techniques go into PCR, and differences in techniques are often related to the type of primer that was used, the DNA that's being amplified, and the detection method. So differences can occur within the PCR process because of these things. So in conclusion, for lack of a better saying, combined PCR allows you to pick one piece of DNA that you're interested in and make as many copies of it as you want. So that's super cool. Like I said in the beginning, you can consider it a genetic copy machine, which I just find super interesting. PCR is just a small part in genetics. It's a very minute, small part. Like genetics is so broad and so huge, and there's so many details that go into it. Like there's just so much to learn about genetics, it's super exciting, and it's really cool because I feel like when you have a better understanding of genetics, you can better understand everything around you and how stuff was made. So this has enhanced my knowledge on PCR, which I'm super thankful for, because throughout the rest of this class and this quarter, I hope that I will be able to better do these labs and assignments to the best of my ability, because now I've enhanced my knowledge so much on this topic. And this is what the class is about. So genetics are what make you you and what make me me, and I just think that's really cool. And like I said earlier, all organisms will have some type of genetic makeup. So through understanding this, you become more knowledgeable and you're able to make the best decisions about the world that you're going to live in. So thank y'all for watching. I really hope that you enjoyed it. I enjoyed making this and learning more about this process. I really feel like I know a lot more about PCR, and I'm so excited to finish this class. So bye, thanks.